Hello and welcome to this MedComNet interview with Dr. Simon Green. Simon joins us as part of our focus on drug discovery for diseases in the developing world. Simon is the Tuberculosis Biology Programme Coordinator at the University of Dundee in the UK. Simon, thank you for joining us.
Thank you, Peter. Thank you for inviting me to talk to you today.
It's an absolute pleasure. So if we just go straight into the questions then. First of all, could you please provide an introduction to tuberculosis and your research conducted at the DDU?
Okay, so tuberculosis is a disease caused by Mycobacterium tuberculosis. It's a lung disease, and its primary symptoms are coughs, sometimes with producing sputum and blood, and you get chest pains, weakness, fever and night sweats, and ultimately it can kill you. Although people think TB is a disease of the past, it's actually the world's leading infectious disease killer, and it kills somebody every 20 seconds. And in 2015, 1.8 million people died from TB. And it's primarily focused in the developing world, but there is also an increasing amount in the Western world due to an increasing amount of poverty. And one of the biggest complexities as well with TB is the increasing amount of drug resistance that is seen. So 500,000 people died because they have resistant TB, and it's not treatable by the standard therapies. At Dundee, we're trying to look to try and develop drugs that target TB, and that's as part of the Tuberculosis Drug Accelerator, which is a collaboration of 8 pharmaceutical companies and 8 research institutes that have been put together primarily by the Bill and Melinda Gates Foundation, expressly to fight TB.
Okay, excellent. And obviously earlier you mentioned resistance and why that is a problem, but with TB, what are the current treatment options and why is it that they are falling short?
So the current treatments were developed in the 1960s and they're old and antiquated. And the current regimen is treatment with 4 drugs for a period of 6 months. And they use 4 drugs. These are isoniazid, rifampicin, pyrazinamide, and ethambutol. And they use the 4 in combination because that greatly increases the chance, or decreases, I should say, the chance of developing a resistance. Whereas if you treat with a single agent, resistance occurs very rapidly. Now, although you treat with the 4 drugs, initially there's a rapid drop of bacterial load in the patient, but there always remains a slow replicating persister population found in specialized bodies in the lung called granuloma. And it's getting the drugs into these regions of the lung and killing these slowly replicating bacteria, which requires the extended treatment out to 6 months. And because of the extended treatment, it means that compliance is a difficulty. Nobody really wants to be taking drugs for 6 months. And because of the compliance, a lack of compliance, that results in the development of resistance because people come off therapy before they're completely cured. And the bacteria that are remaining, there's an increasing level of resistance in the bacteria that remain.
Okay. And then sort of carrying on from that, do you have an idea of what form a novel therapy for TB may take, or are there any treatments you're feeling particularly optimistic about?
So there's been a huge upsurge in the amount of research that's being done on TB at the moment. And I think it's too early to tell which novel therapies are going to actually work. But under the resurgence, there's a huge amount of novel targets that are being pursued. And these haven't been validated in the clinic, but the drugs are now getting to the stage where they will see getting into patients to see whether they actually are able to give clinical benefit. And in addition, there are a number of new drugs as well that are coming along, which are modifications of old drugs, and with better pharmaceutical properties. And these are more likely to, or these have a good chance of working because they're already an modification of an established therapy.
All right, excellent. And As part of the research conducted on TB at the DDU, you collaborate closely with the international TB biology experts and pharmaceutical companies such as GSK, Eli Lilly, and AbbVie. Additionally, you're also a part of the International TB Consortium, sort of as you mentioned earlier. You have support from the Bill and Melinda Gates Foundation and also the Wellcome Trust. How important is this collaboration to TB drug discovery?
I think this collaboration is extremely important. It's, as I think I mentioned earlier, it's a groundbreaking collaboration. The fact it's bringing together the industrial mind of drug discovery companies with the TB experts that are present in academia. And because of this, bringing them together, it's actually transforming the way drug discovery is done, and particularly aiming it for TB, which the modern techniques of pharmaceutical drug discovery with the TB biology, marrying them together to have the best chance of getting new novel drugs. And also with the academic groups that are in the TBDA, they're doing novel work as well to produce new techniques to better understand the drugs that are being developed and how those drugs penetrate the relevant regions of the lung where the disease is infected, so how the drugs get into the granuloma. That's been done by Véronique Dartois at Rutgers. And if you can then modulate the pharmaceutical properties of your molecules so they have better capabilities to penetrate these disease lesions. And there's also other groups such as Cliff Barry at the NIH who are developing marmoset models for looking at how the disease develops, because the marmoset reflects the human disease much better than any other animal. And so therefore you can see how the disease develops and how new drugs potentially eliminate the disease in these models. So I think it's key for TB drug discovery.
Okay, excellent, great answer. And now for my final question. In an ideal world, if you had unlimited funding, what sort of research would you want to carry out and why?
So at the DDU, we are primarily focusing on chemistry and the ADME properties of a molecule. We do very little biology from a TB perspective, because to work with TB is a class 3 pathogen, and you require special laboratory facilities as this is an airborne disease, and therefore you can't work with it in normal labs. So although we work with the TB experts, and that's going very well, we send all our samples out to the TB experts to do work on it. We've seen from the other projects we have at the DDU, so working on malaria and leishmaniasis, that if you have the two projects working, the biology working alongside the chemistry, then that greatly increases the efficiency of the drug discovery process. So if we had unlimited money for the DDU, then I would be interested in building a Cat3 facility here for doing TB biology.
Okay, excellent. As I said, that was my final question. So I'd like to thank Simon for his time, and also to remind our viewers that MedChemNet is on Twitter at MedChemNet, and we'd be absolutely delighted to hear your thoughts on today's interview. Simon, thank you once again for your time.
Thanks very much for speaking to me, Peter.
Right, thank you. Bye.